Benjamin Baram, and today I'll be presenting on using potassium chloride to offset soil salinity and the growth of Brassica rapa. To review some literature, it's first important to note that ocean sea levels have been rising. In the past century, ocean sea levels have risen by an average of 25 centimeters, and estimates already suggest in the next 50 to 70 years, oceans will rise another 72 centimeters. These rising sea levels have big implications for the soil we use to grow our food. Already, 33% of irrigated land and 20% of all cultivated land have already been degraded and affected by high salt levels. To further review some literature, high salt levels stunt plant growth. It's important to note that in plants that are trying to grow in a highly saline soil, crop yields could be reduced by up to 50%. Potassium plays an important role in human salt regulation as well. In high sodium diets, increasing potassium intake can reduce the negative effects. Potassium is also necessary in the adequate growth of plants. High levels of potassium that plants need are essential for the growth, regulation, and daily function in plant growth and their metabolisms. There also does exist a relationship between salinity and potassium. Increases in the salinity of a plant's water source also decreases the potassium levels in a plant's leaves. The purpose of this experiment is to demonstrate if increases to the potassium that a plant is exposed to reduces the negative effects that come from a high sodium content in its soil. It's already known that potassium regulates salt levels in humans, and that in plants, increased levels of sodium in a plant's water source does result in reduce in the potassium content of a plant. This study hopes to establish potassium chloride as a chemical mechanism to help plants grow in more saline environments. The hypothesis for this experiment was that the external application of potassium chloride to Brassica rapa would reduce the negative effects that come from growing in a saline soil. This was thought because of the relationship already demonstrated between sodium and potassium in Brassica rapa and the role that potassium plays in plant growth. This hypothesis was tested by the external application of potassium chloride dissolved in water to Brassica rapa seeds as they grew and matured. The independent variable was the concentration of potassium that the plants would come into contact with. The dependent variables was the total plant length, root length, and leaf size. The controls was the type of plant used, type of soil, salt concentration, and the container size. The methods used for this experiment was to drain a 10 gram per liter solution of salt through 31 plastic cups that had already been filled with soil. These cups were then placed underneath fluorescent lights in 2 plastic trays, and each cup was then planted with 10 seeds of Brassica rapa. Every day, half the plants were sprayed with purely water, and the other half was sprayed with a potassium chloride solution of 10 grams per liter. The plants were then allowed to grow with these daily cycles, and then results were recorded at the end of the experimentation. The needed materials were sodium chloride, potassium chloride, soil, 310 Brassica rapa seeds, 31 plastic cups, and water. So what data can we find from this? The first was an average plant size. The total plant height decreased with the application of potassium chloride by an average of 24.4%. As well, the average leaf size had some decreases as well, decreasing by 42.53% for plants that also had the potassium chloride application. There was a slight increase in the average number of germinated seeds for those treated with potassium chloride, but that was only 1.186%, which is far within the margin of error. So what results can we draw from this? First, it's important to note that the usage of potassium chloride could possibly yield a higher germination rate, but that has yet to be confirmed. Total plant size and leaf size did have drastic decreases in total size, which is also important to recognize. One trend that was noticed is that among the potassium-treated plants, potassium chloride was noticed to be excreted from the leaves, suggesting that the negative effects recorded were directly caused by the high levels of potassium chloride being applied to the plants. The conclusion that can be drawn is first and foremost that the initial hypothesis was flawed. The regular daily applications of potassium chloride did not yield plant benefits, but what should be considered is that this research does not discontinue potassium as a potentially beneficial element for the growth of plants in saline environments, but instead the way in which it was applied to plants does not work. Some errors that should be considered for future research was first that access to better regulated settings, more time, a greater number of plants to grow at a single time, and another set of plants that were not treated with sodium or potassium chloride at all for greater comparisons. Further research in this subject should attempt to account for the extra chloride content of the sodium chloride and potassium chloride, as well to see if different ratios of potassium chloride to sodium chloride would produce better results. Here's all the references cited in this PowerPoint, and all photos were taken by me, Benjamin Baram. Thank you for your time.